Let's talk about endocrine pancreas in this lecture. The pancreas is shown here. The head, the body, the tail, this is the free end, this is pancreatic duct carrying pancreatic juice to the duodenum, which is rich in digestive enzymes as well as bicarbonate. So that deals with digestive function. The smaller percentage of tissue, that's only about 2% of the tissue within the pancreas, are these blue areas called islets of Langerhans or the pancreatic islets. They are the ones that secrete hormones. Well, a section of the pancreas is shown here. You see the acini, pancreatic acini that are able to make digestive juices, and also the enzymes in the bicarbonate. Whereas here, this section is showing an enlarged view of the islet of Langerhans. Here you see endocrine cells. The ones that are shown here are alpha cells and beta cells. There are others that are not shown. They're all listed in the table. So we'll go through the types of cells and the types of hormones they make. Beta cells are the ones that make insulin, important for you to remember. And alpha cells make glucagon. These two both are important in keeping, regulating blood glucose levels standard or normal, okay? Then you have somatostatin, that is secreted by delta cells. They have an inhibitory function on the secretion of both insulin and glucagon. Okay, anytime you think about somatostatin, it is an inhibitory hormone that inhibits many functions, including the secretion of insulin and glucagon. The pancreatic polypeptide is secreted by PP cells, and that is important in the regulation of appetite. So these are the four types of endocrine cells and four different hormones, all, as you can see, are protein hormones. Large proteins, these are amino acids of the long chains in a three-dimensional arrangement. Now, we will focus on insulin first, and here is the structure of insulin that's shown. You're talking about its synthesis beginning as a pre-pro insulin. You have two chains here, chain A and chain B. Chain B is in red, okay, and chain A here, this is yellow, and chain C here. So what happens is, this is all additional structures that have to be removed, and that is where this is being modified. And note here, proinsulin is where you have lost the signal sequence, that is removed, and the rest is still around. And then when you go here, what you're talking about is the removal of this excess portion, and you only have the chain B and chain A together. Okay, so these two together form the insulin structure. All right, so that is a quaternary structure, meaning when you have two polypeptide chains come together to form a protein, it's a quaternary structure. Now, so that is insulin structure. You have two polypeptide chains, and pre-proinsulin to proinsulin to insulin is how its synthesis happens, and the hormone is typically stored in the vesicle and released as needed. Before we talk about the release of insulin, we want to talk about the different types of transporters that exist in the body. Okay, some depend on insulin to transport glucose, some do not. And that's where you need to know which ones do and which ones do not. GLUT4 is, GLUT stands for glucose transporter. 1, 2, 3, 4 are the ones here. GLUT4 is the one that's insulin dependent. That's present in the adipose tissue and skeletal muscle. And note that GLUT2 is present on many cells. Liver is an important one for you to remember, having GLUT2. Okay, beta cells have GLUT2, and the renal tubules. So those are some important details for you. GLUT1 in some tissue, fetal tissue in the blood-brain barrier, GLUT3 in the neurons in the placenta. So none of those depend on the insulin, but GLUT4 does. That means adipose tissue and the skeletal muscle depend on insulin for these cells to utilize glucose. That's where it's important. Skeletal muscle makes up about 60% of the body's mass, and then if these cells depend on insulin to use glucose, if glucose transporters are not working, that's what you call as insulin resistance, then you won't be able to utilize glucose that's in the blood, and that's when hyperglycemia occurs. Okay, here is a nice summary of how insulin is released by the beta cell. So this is our beta cell. You have GLUT2 here, which is not dependent on insulin. So after eating, glucose levels begin to rise, and some glucose gets into the beta cell. So as glucose arrives, it's used in the metabolic pathways, and what you see is the glycolysis and Krebs cycle and the electron transport system releasing ATP. So certainly you will begin an increase in the levels of ATP within the cytoplasm. Okay, so this story is hopefully clear. Glucose enters the beta cell and it's utilized in the energy producing activities, and energy is produced, and this ATP would close a channel. It's a potassium channel that's referred to as ATP sensitive potassium channel. So potassium channel that is sensitive to ATP would close. This is where you have potassium not leaving the cell. So that would affect the membrane potential. The membrane potential will not be negative 70 anymore, but instead it would be a lower number, negative 60, negative 50, etc. So this is how the cell becomes depolarized. Depolarization then leads to the opening of a new channel, voltage sensitive calcium channel. So this channel allows calcium ion to enter the cell. It's like in a synapse where you have the neurotransmitters stored in the vesicle and are all in a synaptic knob. Calcium has to be there to move these vesicles towards the cell membrane. That's exactly how it works within the beta cell. So you have calcium ion within the cytoplasm, and this is the one that literally moves the vesicle that's filled with insulin towards the membrane. The membrane and the vesicular membrane both fuse, and at some point the membrane dissolves, releasing insulin. So these are the steps involved in the release of insulin. What triggers the release of insulin is where you have to know that there is glucose. The levels of glucose is higher in the blood, and some of it makes it to beta cell, and that is a stimulus for insulin to be released. So as you eat, your glucose levels raise in the blood, and that is how insulin secretion begins. Okay, so here is the summary of the action of insulin. So insulin is released. And how does it bring about its effect is what we're going to understand in this slide. So you have an insulin receptor. It's a very unique receptor. Okay, it has two subunits, alpha and beta. You could consider these as two subunits. Okay, The subunit has a portion within the membrane as part of the cytoplasm as well. And another subunit here is the one that interacts with the hormone. Okay, hormone binding is number one. Okay, binds to the receptor. The two subunits come together. That is when the activation of the receptor occurs. Another unique thing about this receptor, insulin receptor, is the portion of the receptor, once it's activated, will serve as tyrosine kinase. Okay, so this is an enzyme combined with the receptor. All right, so now insulin binds to the receptor, the two dimers will come together and activate the receptor, and then there is tyrosine kinase activation. You see, and it provides a site for phosphorylation. So now the events within the cell vary from one type of cell to another. Is it the liver cell? Is it the skeletal muscle fiber? Is it the adipose tissue? Depends. So you need to imagine what could be one of the functions of insulin and think about that. If there is glucose that is entering this cell at the same time as insulin binding to its receptor. Okay, so insulin is present when there is increasing levels of glucose. That means there is transportation of glucose into the cell. It could be GLUT4. Okay, because now that there is insulin, GLUT4 is able to move glucose into the cell. So once it's inside the cell, it is the activation of steps where glucose is converted into glycogen. This is the storage form of glucose. So you notice here, glucose is now converted into glycogen and stored within the cell. That's how insulin brings about its action. All right, so if there is glucose, then it could be converted into fatty acid, and eventually it is converted into a lipid. Okay, so those are some things that are shown. What if there was amino acid entering this cell? It could be converted into a protein. So these are some effects that are shown in this slide, but there are many more events that insulin could turn on. All right, so now let's talk about a number of factors that could influence or interfere with the insulin release. So we talked about insulin release by beta cell. What are some things that would promote the release of insulin? What are some things that inhibit the release of insulin? Increased blood glucose, increased blood amino acids automatically trigger the release of insulin, which we learned by looking at the beta cell and how glucose entered the cell and triggered the release of insulin. Also note parasympathetic stimulation. Parasympathetic stimulation is true during digestion. So digestion, activation of the parasympathetic system, release of insulin. So they go together. These events are connected to one another. So you need to know that if parasympathetic activation increases insulin release, sympathetic activation would inhibit the release of insulin. That's what is shown in this step. Decreased sympathetic activity would decrease epinephrine, decreased insulin secretion. Okay, so then the nitric oxide is also supposed to stimulate insulin secretion. This is another neurotransmitter, and a decreased plasma potassium ion would decrease insulin secretion. Okay, so those are some events that would affect insulin secretion. Okay, so now this is the summary of what all insulin does at some major cells. In most cells, you know how glucose gets into the cell and it's utilized for